Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 50, and it's going live on March the 13th. 2 straight months with a Friday the 13th. It is World Sleep Day and it is Dermatologist Day, and we do have kind of 2 themes on the podcast this week. It was International Women's Day last weekend, and this week it was World Kidney Day, so our interviews reflect both of those. This weekend is Mother's Day here in the UK, but not in North America, so my wife expects it to be honoured twice. And it's also that awkward time of the year when clocks have sprung in some parts of the world, but not in others, which always throws off setting up interviews. 1 o'clock? Okay, well, 1 my time or 1 your time? Oh, and then, okay, is that now 12 with the time change or 2? Or now that we've changed back as well, is it 1 still or 12 or 3 o'clock? Last week we talked to Hasib Ahmad, CEO of Pure Spring Therapeutics, about gene therapies in kidney disease, and we chatted to Pahini Pandya, Panakia CEO, about colorectal cancer biomarkers, among other things. This time it's a bumper 3 interview episode. The conversations this week are with Hansa Biopharma CEO Renée Aguiar-Lucander about Imlifidase for kidney transplant patients. And we chat with Kyron.bio CEO Emilia McLaughlin about glycosylation, and we discuss hydrogel-based drug delivery with AmacaThera's CEO Mike Cooke. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which thankfully lots of people are doing. That's also the address you can use if you're interested in advertising with us, which unfortunately not a lot of people are doing, or sponsoring a podcast episode, or for event coverage. Alright, let's take a look at the news you may have missed over the past week at deeptechdigest.com. We had an article on a new approach to drug development. Now BIOS and SGS are partnering to manufacture an RSV challenge agent, and a drug to treat advanced squamous cell carcinoma of the anal canal has been approved. The first reliable biomarker test for early kidney transplant rejection detection in living donor recipients has been developed. A study shows AI boosts breast cancer detection by more than 10%, and a collaboration is hoping to advance novel combination therapy for glioblastoma. A new company is targeting dry eye disease with a first-in-class therapy. An atopic dermatitis treatment has been approved, and a new mass spec platform has been launched. A new prostate cancer treatment has been given approval, an anti-cancer drug has been given the go-ahead by the FDA for a phase 1 trial, and you can read all of these and plenty more at deeptechdigest.com. Okay, time for the interviews, and first up, everything you needed to know about glycans in a conversation with Kyron.bio CEO Emilia McLaughlin. Kyron.bio's glycobiology platform aims to enhance the efficacy, safety, and scalability of next-generation antibody therapeutics across multiple disease areas. All right, hi Emilia, thanks so much for being with us today. Perhaps first you could tell me a little bit about the company.
Yeah, so Kyron.bio is a glycan engineering company. So glycans are sugars that are found on the surface of therapeutic drugs, and they're found on 2/3 of the proteins inside the human body, and they're also found on the majority of biologics that are on the market. What we do at Kyron.bio is we precision control these glycans. So we work on N-glycosylation, and we control how long the branching of the glycan is and how wide the branching of the glycan is. So we really precision control what that sugar looks like that's found on the surface of a therapeutic drug.
What was the connection between, you did your PhD there in
Yeah.
Paris and you started a company. What was the connection? How did you kind of make that leap from one to the other, I guess?
Yeah, that's a really good question. So I studied, rather unusually, I studied parasites during my PhD. That's what brought me over to Paris. I'd previously been looking at malaria parasites, and I came over and did a PhD looking at African sleeping sickness. And the parasites I studied have a very interesting modification to their surface coat that's a glycoprotein. So they're really glycoprotein factories. And it's just another example of how glycans crop up all over biology, across all domains of life, and have different functional benefits, across viruses, parasites, human proteins. There's a huge, enormous array of ways in which glycans are used across biology. So that got me looking at glycosylation. And then after finishing my PhD, I joined a tech incubator called Entrepreneur First. And then that kind of was the foundation of the company. And that was when the company was created 4 years ago. They were the first investor into the company, but it sort of led me on this path of how can glycosylation be harnessed for good in terms of drug design, and what could that open up in terms of potential for our biologics.
Could you maybe tell me a bit more about the relevance of glycans in therapeutics?
Yeah, so glycans are very important for multiple parts of a protein functioning. So they're important in the stability, where it's targeted to, how it interacts with the immune system, how that drug is filtered, so the half-life and the PK. And there are some examples of how glycans have been used in the past in drug design. So in antibodies, all antibodies have a core glycan on the FC297 position. And it's been shown that when you remove the fucose from that N-glycan, which is just one really small element of it, you can massively impact the ADCC activity of that. So that's recruitment of immune cells to a tumor. And that's not a minor tweak, it's an entire basis of certain drugs in terms of recruiting immune cells to the antibody therapeutic. And that's just one example of how a glycan can be modified to interact with the immune system to have an enormous impact on the way that biologic is functioning. But to date, what we've done with glycans has been really limited to that low fucosylation on an antibody FC297 position. And what we're doing at Kyron.bio is we've built the tools to really fully control the entire length of a branching end glycan. And that opens these glycans up as potentials for drug design. And that opens a lot of new avenues in terms of how we can harness glycosylation to deliver better therapies for patients.
And so why is it an area that's been limited so far? Because if it's clearly got lots of potential, then why hasn't it been tapped into yet?
Yeah, so it's a combination of things around. So glycosylation is detected by an analytical method called mass spectrometry. And the analytics until now has been relatively limited. It's much more laborious looking at the glycome and doing mass spec on glycoproteins compared to sequence analysis, for example, of a protein sequence. And we've had some recent advances in mass spectrometry that have allowed us to be able to define much clearer which glycans are found at specific sites. So not only their presence or absence, but what exactly do they look like? And there's also advances in engineering of cells. So we work at Kyron, we engineer cells to control the glycosylation pathway. And those gene editing advances, allowing us to layer on different combinations of gene editing in parallel, have also been critical to that. And then I'd say the third element is around AI and having tools that can allow us to better understand how, if we modify certain positions of a protein, particularly with a glycan, how does that impact the drug functioning, target binding, and other impacts that those glycans have on the stability of the drug as well.
So what does that mean for drug discovery and for drug efficacy?
Yeah, so we've already shown that using precise glycans positioned on an antibody therapeutic, you can prevent unwanted immune attack. So when I say unwanted immune attack, I'm talking about anti-drug antibodies that are raised against a therapeutic that come and clear the therapeutic. What we've shown is you can strategically place glycans to prevent that unwanted clearance. And we've shown that in patient samples across multiple therapeutics. And that's a really interesting example of tangibly what do these glycans mean. But we don't stop there. These glycans have enormous potential in terms of the PK, in terms of internalization of a therapeutic, targeting of a therapeutic, and the overall stability of a drug, which translates to the administration method. So whether a drug is going in intravenously or whether it can be subcutaneously administered. So there's quite a broad amount of things that glycans can impact. And at Kyron, we're only beginning to scratch the surface of understanding all of the kind of incredible impacts on these therapeutics that controlling the glycosylation can have.
And how does that impact drug design? I mean, are you able to make different changes for different conditions and different drugs?
Yes, because we have a toolbox where we can say exactly what type of glycan we want at a specific position. Different glycans are associated with different functions, and we've known that for a long time. Glycobiologists have been working on this for many decades. And so what we're now able to do is say, okay, if you want to, for example, target a specific type of cell using a specific type of glycan, that can be positioned on using our technology.
And so how does the process actually work? How do you do it without giving away any trade secrets? How do you do this?
Yeah, so we work in genetically engineered CHO cells. So CHO cells are the cell line of preference for the industry for antibody therapeutics. And we work in industry standard CHO cells that have been genetically modified to control the glycosylation pathway. And so there's genetic engineering that has happened to those cells. And what we do is we combine that with engineering of the antibody therapeutic itself, introducing in specific N-glycan sites at regions which we have detected to be particularly beneficial for different functional benefits. So it's a combination of engineering of the cell line and engineering of the antibody therapeutic to introduce in novel N-glycan sites. And we then express that modified sequence in our cell line of choice. So if we want to, for example, have a sialylated N-glycan at that site, we express it in our sialylated cell line.
What does this mean for drug discovery? I mean, where is it going to have impact? And does it mean that you'll be doing clinical trials or working with companies on their clinical trials?
So we're doing both. We're partnering with companies, and we just recently announced a partnership with Servier, French pharmaceutical company, to work on an antibody therapeutic together, and we'll also be advancing our own therapeutics towards the clinic as well. So it's a dual model. We're a platform technology with asset development also ongoing.
It sounds like it's something that, because it's not really been done and it has a lot of potential, do you think that this is something that could radically transform the industry? I know we hear the word disruption all the time and I hate it, but do you think that there's that capacity?
Certainly. At Kyron, we like to think of glycosylation as the fourth dimension of biology. So far we've said DNA makes RNA, makes protein. But the fourth dimension of that is these post-translational modifications, and glycosylation being a critical one of those in terms of its broad functionality in the body and what that can do for biologics. And I think that is a really disruptive technology, like having the tools to be able to control glycosylation, which we haven't had until now, has enormous potential in terms of disrupting how we build our biologics and how we view their design and how we can integrate precise N-glycans into the design of biologics.
We're talking about glycosylation, but are there any other processes that could similarly be manipulated?
There are other post-translational modifications, of course, but the one we see as having the biggest impact is definitely glycosylation in terms of drug design. Yeah.
And how are you able to ensure consistency, especially at scale?
This is a really important part of what we work on, because we know it's very important for any partnerships and also for the FDA to see that glycosylation is consistent. So that's really the core of the technology, is being able to show that we have consistent glycosylation. Simply, it's achieved through genetic engineering that we do on the cells, but what we've been able to show is that the glycosylation remains very precise, meaning over 97% homogeneity of the N-glycan site over 60 generations of cell passage. And we also do scale up in bioreactors to show that not only does this work in a shake flask in a small incubator, but it also works when we go into a large industry relevant bioreactor as well. So the technology, it needs to be able to scale. So that's something that has been a major focus of ours within the company.
And you mentioned the partnership with Servier. Could you tell me a little bit more about that?
Yes, in 2024, we won their Golden Ticket Award. So they select one startup every year that's a laureate of their Golden Ticket. And this really accelerated the relationship with Servier. So they've been following our progress, and we've been working side by side for the last 2 years. And I think they're real believers in the potential of glycosylation to transform drug design. And they're also very interested in ways to develop better therapies for patients. And we have that shared vision. And so it's quite a natural partnership that evolved as part of that. And really happy to be working alongside them on glycan control for an antibody selected of their choice.
And are there other partnerships that you have as well, or are you still actively looking for partnerships?
We can't currently communicate on other partnerships, but we are open to partnerships with pharmaceutical companies at the moment, and happy to discuss more with those that are interested.
And what does it mean ultimately for patients? Does it change drug delivery, or what's the end result here?
So what this opens up for patients is new treatment opportunities, so treatments that are more effective, that last in the body for longer, that can be dosed at more favourable frequencies. It also opens up potential for new targeting, so new diseases that can be treated using biologics.
One of the biggest problems that needs addressing is the fact that some people just don't respond and other people do respond. Is that something that you'll be able to address?
Exactly. And that's something that we've already shown in our initial patient studies that we've done. We've already shown we can address those patients that are refractory to current treatments.
So what do you think that the future holds for this area of technology? Do you think it can go even further than what you're doing now?
Yeah, I think there's enormous potential. And I'm always, there's only a handful of companies working in this space at the moment. And I'm actually always happy to see when there's other companies in the glycan space cropping up, because I think we're really just at the beginning of scratching the surface. There are companies that turn this on their head and they target glycans on cancer cells. I think that's a really interesting area as well. And with glycomics and advances there, we're better understanding what we can target in terms of N-glycans and what are stable targets. But I think that there is enormous potential in this field, and we're only really beginning to see the benefits now.
You mentioned there's only a couple of companies. Why is it so underserved, I guess?
So I think those reasons that I mentioned earlier around the analytical techniques being very laborious and challenging, we're getting much more high throughput in how we can analyze N-glycans, and there is quite a bit of innovation in that space. And I think that's limited things. There were also some early challenges in the space with glycan-engineered yeast cells, for example, in the early 2000s that proved challenging. And so that's kind of put people in the industry off. Also, I think that glycans are historically a bit of a headache for people in drug design. They're there, they're present, but we can't control them. And you've got to report them to the FDA because they're so important in how your drug interacts with your patient's body. And so people have tried ways through process optimization to control them, but it's really been something that people are looking to manage rather than thinking of that question in the reverse of like, hey, how can we harness these in drug design? So I think there's a couple of confounding factors there. But I do see the tide starting to change. And for example, this partnership with Servier shows that they are very willing to embark on glycan engineering in their therapeutics.
Obviously, there are lots of opportunities, but are there still challenges involved?
Of course, every startup, we're a startup. Every startup has challenges. We're a small team. And yeah, of course, there are many, many challenges that you face day to day. I think what's really important is always listening to the customer, and the customer changes depending on what lens you're looking at it from. But that could even be so far as the patient. And I think we should really keep patients in mind in terms of what they need in terms of improved therapies. And sometimes the customer for us, when we're looking at through the lens of partnerships, is the pharmaceutical company as well. And what's been consistent throughout for me is that you need to have both of those lenses on what do patients need and what a pharma company is looking for, and what's also going to integrate into a pharma company's existing system. There's no point in us developing our technology in a cell line that a pharma company has never used before, would not like to put in a bioreactor, and would have to, invest millions and millions into developing new infrastructure for. So we knew from day one that what we have to do has to be in a regulatory standard CHO cell host, for example. And then when we're looking at targets and drug development, it's really looking at the patients and the clinicians and what are their challenges currently in the clinic. So I think those are the kind of ways that I navigate that. But of course, there are always challenges in running a small startup, but they're exciting, and that's where the learning happens as well.
And what is the next, 12 to 18 months look like at the company? What are you kind of working on?
So scale up is a big focus of our work at Kyron.bio at the moment. We're also expanding out our in vivo studies to demonstrate the safety and efficacy of our glycan engineered therapeutics across different assets that we're developing, and of course expanding our partnership pipeline as well.
So now you know about glycans and why Kyron.bio is hoping to make a difference for patients and make a huge splash in the life sciences industry. Next, if you're going to administer a drug, you'll need a method of delivery. AmacaThera is the developer of a next-generation hydrogel-based drug delivery solution. So to tell us about it is the company's CEO, Mike Cooke. Mike, thanks a lot for taking the time to talk with us today. Looking forward to hearing all about it. Could you maybe give us a bit of background on the company first?
Yeah, so AmacaThera, it was founded by myself and Molly Shoykad out of the University of Toronto. And really what it's focused on is developing this novel hydrogel platform and advancing it into the clinic. So we've had some really cool data in the lab where we could show this material was great for drug delivery. And AmacaThera is really focused on how do we take these great inventions and transform them into products that can be used in patients. So lead asset is focused on post-operative pain. So how do we manage the pain locally and decrease the need for opioids? And then from this, we're sort of building out a suite of platforms on our platform where we look for different applications of the technology. So where else can this be applied? So really, in a nutshell, AmacaThera is a drug delivery company with this novel hydrogel material looking for applications of the technology.
Just out of interest, where does the name come from?
Yeah, so the core tech, it's made of two polymers, hyaluronan and methylcellulose. So in the lab, the core material always used to be called HAMC, H-A-M-C. My mind, that's a little bit illiterate, but that was what the nickname of it was. We actually incorporated as HAMC Therapeutics the same day we incorporated another company incorporated in North Carolina. So legally it was fine. We were in Canada, they were in the US, but it caused a lot of confusion. So we changed the name. So Amaca actually means hammock, I think in Italian, and then Thera for therapeutics.
Could you tell me a bit about your hydrogel platform and how that works?
Yeah, the core tech, it's these two polymers, hyaluronan and methylcellulose. So the polymers, the properties that they have are shear thinning and inverse thermal gelling. So when you think of shear thinning, think of ketchup. Ketchup doesn't flow until you squeeze the bottle or smack it. When you do that, you're actually applying shear, and that material is thinning. And then when the ketchup's on your plate, the shear is removed and holds its shape again. So you have this material, you can get it out of the bottle, and it will stay where you put it. Like ketchup, it holds its shape. It's not like water. Put water on your plate, it'll just cover the plate. So that property is called shear thinning. Second property is inverse thermal gelation. So think of Jello. When you make Jello, you heat it up, and as you cool it down, it forms a gel, as is the inverse. So as you take it from room temp to body temp, it forms a gel. So inverse thermal gelling. So taking together these two properties means you have a material that can be injected, and it will stay where you put it. So it's a pretty simple material, right? It's made of these two polymers, and the core to it is like really just localizing therapeutics to where you inject them.
Could you tell me what this technology is applicable to?
Yeah, so what we've done is when we first founded the company, we went and spoke with the surgeons and the anesthesiologists, and we spoke with about 100 surgeons and anesthesiologists to really say to them, like, we're trying to develop a product that can be used in the operating room. What is the unmet medical need, and what do you need? And this was back in sort of 2016 time. At that time, they're like, look, the opioid crisis is a real thing. This is going to be an epidemic. We need help with this. So really what we did was we tooled the tech to first address that. So how can we take the anesthetics that are used right now, localize them to an incisional site, improve the efficacy, decrease the need for opioids? So really laser focused on building a product on the platform. But the platform itself, it's applicable to really a wide range of different applications. So we have data showing stem cell transplantation. So some of the early R&D data in the lab was stem cell transplantation for stroke. So taking neural stem cells, injecting them to the brain, and then watching recovery of mice. So there's like really cool applications. We have applications for small molecules, proteins, antibodies, growth factors, peptides. It's a pretty exciting material because of the broad compatibility.
Is it something that other people are working on? I mean, are there any similar products to this or being developed?
Yeah, so it's interesting when you think, because it's a platform tech, right? So you can look at it from two perspectives, right? Look at it from a product perspective and a platform perspective. So really the products we develop on the platform are highly differentiated. So like the lead asset for post-operative pain, right now there is no technology that can do what we can do. So looking at the specific product is really differentiated. What's exciting about the platform though is this broad compatibility and this wide range of different drugs we've shown you can combine with that material. There are other platforms, but they tend to be more niche-y, so they like just work for a subset of molecules. Our tech really works for really a wide range of molecules, which is pretty exciting because we validated the product with the small molecule lead asset, and now there's multiple discussions with other pharmaceutical companies around other applications of our technology.
And so what are the advantages over existing products?
Yeah, so when you think about the pain product, right, specifically, right? So right now, the majority of the pain control is used like using solutions of anesthetics. So very similar to the anesthetics you'd get when you have a cavity or a dental surgery. The freezing lasts a very short length of time, like it's like several hours is sort of what this lasts. So you're taking these drugs, sort of freezing the incision for several hours, they're wearing off, and you're taking opioids. So what we've done is we've taken the same anesthetics, put them in a gel, you keep them locally, you keep that local concentration, and you keep the drugs where you want them. Other technologies, so Pacira, actually, that's our partner with the lead asset. They have another tech, which is a liposomal bupivacaine. There's 2 approved sustained anesthetics on the market. One is Pacira's product, and that's really the market leader right now. Where we believe the advantage of our technology is providing a longer duration of efficacy. So their product works really well, but we think we've got something that will last longer, and that was why we partnered with them on that. And then sort of just coming back around to thinking about the platform and how does the platform differentiate from other platforms. This is going to sound kind of silly, but one of the key things to this is the simplicity. Just 2 excipients, 2 polymers in there. Very straightforward, very easy to manufacture. When you look at like healthcare prices and cost of drugs, everything is escalating, right? And in today's market, you really need technologies that are cost effective to manufacture. And that's really what we developed. Like we've developed a very simple, I don't want to say cheap, but I'll say cost-effective material that can really give like some differentiating features to these drugs. So yeah, it's the beauty of our tech, I would say, as a platform is in the simplicity.
Could you tell me about the licensing agreement that you have with Pacira?
Yeah. So we licensed the lead asset, so that's the pain drug to Pacira. We signed the deal in November. Pretty excited to work with them. They're a fabulous group. They're just great people to work with. They're super supportive, and there's a huge benefit to both companies. So we're pretty excited by this collaboration. So the deal, it was 230 million, sorry. There was 5 up front, and then Pacira's covering the development cost of the asset. And that's really the exciting piece, right? So as a small biotech company, like AmacaThera, it's a relatively small biotech company, having a partner that can help and support and cover the cost of the development, that's a huge win for us. So far it's been a great partnership. We've been working hand in hand to develop this. We're currently working on some of the manufacturing, so manufacturing clinical trial material. We're lining up clinical trial sites to start this year. So within 2026, we want to have dosed our first subject in our bunionectomy trial. So yeah, just pretty excited to have them and to have their support and to be advancing this asset. And Pacira, they have another product on the market. It's a sustained anesthetic. They've been there. They've done this before. They really know what they're doing. So it's a fantastic group.
And could you tell me a bit about AMT-143 and what that is and how that works?
Yeah, so AMT-143, that's the nomenclature now for the pain asset. Eventually, hopefully when we're approved, we'll give it some sort of cool name and make it sound good. But for now, AMT-143 is the boring science name. So the cool thing, some of the differentiating features about what we've developed is this really cool formulation work. Again, very simple, that was done with the release kinetics of our product. So right now, the other products, you have to take a vial of a solution of the drug and mix it with the long-acting anesthetic. And that's so you have a rapid onset followed by a sustained release. We actually formulated our technology so it has a rapid onset followed by a sustained release. So from a single injection, you can get now this rapid onset and sustained release, whereas the products on the market, what you have to do is you actually have to take 2 drugs. So you'll take a solution of the drugs, you'll inject that, and you'll take a long-acting and you'll inject that. So we really made this differentiated profile. And then the other thing that we've done is we've been able to push out the duration of efficacy. So if you look at the currently available products, you have solutions of anesthetics such as Marcaine lasts several hours. Pacira's product is around sort of 2 or 3 days of efficacy, and then we're pushing this out way beyond that. So in humans, we saw around 14 days of pharmacokinetics. So we're looking at drug levels in the plasma, could show that we're having sustained release out to 14 days. And then in some model systems, we've shown beyond 7 days of efficacy is what we've got. We believe really if we can just extend this sort of 2 to 3 days to 5+ days, you're going to have a game-changing product. If you think about like post-operative pain, sort of a bit broader and really what's happening in the environment, right? It's hospitals have this huge push to get people out quicker. So they need to get them out, reuse the beds. So if there's a way to manage the pain better and get people out, get them sent home, they're happier, you can reuse the beds. And the other thing is ambulation. So after these surgeries, one of the things is you've got to ambulate. So you have your knee surgery, get out there, get ambulating as soon as possible. And studies have shown that helps recovery. So if you can make people more comfortable, you're actually going to have a huge improvement on their recovery. You're going to have a huge improvement to the healthcare system by reducing costs.
And I know there are other non-opioid pain medications in existence and being developed currently. What are the kind of selling points, I guess, for what you're doing as opposed to what else is out there?
Yeah, really, the products that are on the market right now is solutions of anesthetics, so Marcaine and Neuropen, and then Pacira's product Exparel. That's basically most of the market. Differentiating ourselves from that is the duration, like really this longer duration. And then a relatively simple manufacturing process should get us to a price point that makes sense for everybody. There are other drugs in development. A lot of them, to be honest, get stuck in academia. And that's something that we did well. We actually got this out of the lab into a commercial entity and were able to advance it, doing some of the more sort of commercially translational driven things, such as the clinical trials, the GMP manufacturing. So there are a lot of the other techs that are sort of stuck in the university, I would say. Some of the other techs that have been quite interesting of late, though, and I think just thinking more generally about pain, and not necessarily about AmacaThera, but Vertex generated a lot of interest around pain recently. They had their NAV 1.8 drugs. They're like spending an absolute ton of money running those massive clinical trials, and I think they got everybody excited and got pain to the forefront of people's minds and got us thinking about pain again and how important pain is, because really everybody has pain. Nobody wants to be in pain. So I think that was a great thing for all pain companies.
Just as a matter of interest, you've mentioned a couple of times about price points and being cost effective. When either medical services or healthcare systems, when they're deciding upon new treatments, how much of a factor is cost effectiveness? I mean, are they going to go with something that's cheaper simply because it's cheaper, or are they going to go with the best because it's the most effective?
Yeah, no, that's just a great question, right? And just thinking about that, and it's just so important. I think the heydays of the healthcare system would pay whatever for the best drug. That's just not a thing anymore. I think all healthcare systems are getting price conscious, right? And they're really looking at the efficacy of the product and doing this sort of health economics assessment of like, does the product, okay, you've got the product to work, does it really work? Does it make a difference? Can we reduce healthcare costs? And I think also when you sort of look at like the US, Canada versus Europe, right? Europe is very price conscious. It's epically complicated because over in Europe, like you sort of think of Europe as one entity, but it's not really, because like, you've got to go to each country and get your drug approved there. And each country has its own system, has its own health economics to that. So I think really if we want to call it the good old days, but I'm not sure we should, of where the healthcare systems will pay whatever. I don't think that's here anymore. And I think you can see that in some of the drugs that are getting approved now. Really, price is a huge driver, and like cost of living is going up, the cost of healthcare is going up, everything is going up. So I think we also, like as drug developers, right, we have a responsibility to be developing cost-effective drugs that are really like improve the healthcare system.
So where are you at on the clinical trial pathway with 143 and with others?
143 is that's the most advanced asset. So as AmacaThera, we completed a phase 1 using the hydrogel alone, so the core tech without a drug. We also completed a phase 1 using the technology combined with the anesthetic, so AMT-143, and now we're working on the phase 2 with Pacira. So super excited to have their support and guidance through this. The phase 2, we're aiming for the first subject dosed this year. That will be a bunionectomy trial. I don't know, maybe a little bit of a aside, but we'll just sort of talk about approvals and how these drugs sort of get approved. Like the first trial is in bunions. Really, we're not looking for a product to treat people's bunions, although they're painful. And really what that is, it's a model of bony tissue, and it's a well-established model of pain that's accepted by the FDA and the regulatory bodies. So really, we're looking at these sort of like model systems to show that the drug works in this. And the plan is if you show you can use the bony tissue and then a soft tissue, so like hemorrhoids, hernia, abdominoplasty, taking these two sort of tissue types together, you can then try and get a broad approval for your product. So really looking, because ideally you don't want to be selling a product into a hospital and the hospital says, oh, it's just approved for knee surgery, it's just approved for bunions. You want a broad approval as possible, and to do that you do multiple trials in multiple different incisions. So that's really the path we're going down with Pacira. They were super successful doing that with their product Exparel, so following a similar path and have their support on that. And then sort of loop it back a little bit to the discussion around sort of health economics and improving the healthcare system. Sort of some of the things we want to look at right during the study is like, are we reducing length of stay? Are we improving ambulation? When we eventually get to the knee studies, we'll be like, are we improving range of motion? Are people, can they move their knees more? Can they ambulate? Can we reduce the number of opioids taken? So if we measure how many opioids people are taking, is that reduced when they're on our medication? Right? Really thinking early on about some of these drivers to show that there's a benefit to the healthcare system.
You've talked a bit about Pacira. Do you have other partnerships?
Yeah, there's a few other partnerships. It's actually pretty cool. So we have a partnership with Merck Animal Health. So we actually licensed the lead asset to Merck Animal Health first for use in animals. And that's pretty cool, right? Animals have pain. Animals can't really tell you when they're in pain. They sort of, you look at them and see they're in discomfort. But the cool thing about our tech is if you can inject it, suture them up, and it's done. So that's a great benefit to an animal. The animal has a surgery. The owner doesn't have to give it pills. They don't have to manage the pain management. The surgeon does that at the time of surgery, sutures it, and it's done. So we have a pretty cool collaboration with Merck Animal Health looking at the application in cats and dogs, so it'll be companion animals. And here again, it's just a great collaboration because they just bring a wealth of knowledge around getting veterinary products approved, which is not our expertise, right? So really they're thinking about how to design the best clinical trial and really how to get this approved. And that fits nicely with the mission, right? So it's also, if people's cats and dogs aren't in pain, people aren't being given opioids for their cats and dogs, we're again reducing opioids that are getting out there in the wild and just really focused on managing the pain at the time of surgery, suture the animal, done. That's one that's pretty cool. And then there's a few other undisclosed partnerships, and there what we're typically doing is taking their drug and combining it with our gel, showing some proof of concept, and then hopefully advancing those towards a license. So there's a few other of those, and we have them around various molecules like biologics, small molecules, peptides. So it's pretty exciting times, and there's a lot of interest in the material. Because,
as you said, animals don't give necessarily a signal that they're in pain. Often people fail to give them the meds, or they have difficulty giving them the meds, and so they don't bother. So this has clearly got a lot of positives in just the whole process of pain medication, I guess.
100%. You're spot on with compliance, right? And people are just as bad as animals, right, for compliance. They don't take their meds. So that's one of the things we're sort of looking at, are the applications where a single injection and then that you're compliant with the medication, right? So 100% agreed.
Is it something that's in the same way that you would continue taking painkillers for a month after an operation or whatever? Do you have to top up that medication, or is it just a one and done?
That's an interesting point. So there's something called chronification, and it's where acute pain becomes chronic pain. So poorly managed acute pain can actually lead to chronic pain. So one of the things that we're going to see is that if you manage this acute pain period right after surgery, let's get the pain under control, will that prevent chronification? Will that prevent these long-term pain applications you're having? We've really focused the lead asset on this idea of injecting to the surgical site, and we haven't sort of looked at the post-operative pain market, but it's a huge market, a huge unmet need. Like probably everybody over a certain age, I know I woke up on one of my birthdays and everything started aching. So I think chronic pain is it's a huge need as well. I think later we might start to look at some applications there because it's huge for everybody. Nobody wants to be in pain.
Do you have anything else in your pipeline?
The company started out, we picked something. We picked post-op pain. We're like, this is the way to go. Spoke to the surgeons, the anesthesiologists, validated that, went down that path. right now we're getting a lot of inbound interest from pharma. So we're actually working on the ideas that they come up with. So they'll come to us that we've got this class of molecules, we need a long-acting injectable, can you develop it? So really the pipeline now is a lot driven by pharma. We are looking at developing some more assets in-house. So we have some oncology drugs that we're pretty excited by. So again, this idea of like, where do you need like local sustained release? So can we inject the drugs, keep them locally, decrease the systemic tox, right? Because like for oncology drugs, everybody knows like these drugs work really well. The problem is they kill all cells. So just taking these drugs and like localizing them, you can even see the systemic issues with hair loss, right? When you take oncology drugs, you have hair loss, and that's basically a systemic tox you're looking at, right? So if we could take those, localize them to a tumor, you'd improve the efficacy, decrease the systemic toxicity. So that's really one area we're interested in. And then the other area that we're sort of developing in-house is sort of just thinking about, again, where do you need long-acting injectables? And then sort of thinking at it from a platform and a product perspective. So what products can we build to show that a tech works for this category of drugs? And there's some pretty cool stuff sort of looking at like drug loading, looking at localization. So yeah, we have one or two in-house ideas, but right now the bulk of the ideas, there's a pull from big pharma for this kind of technologies.
And we haven't mentioned winning an award, but what's the significance of the Life Sciences Ontario Emerging Company of the Year award? What does that mean?
Yeah, no, that's pretty cool. So yeah, we won the Life Sciences Emerging Company for 2025. So we're pretty excited about it. It really validates the company. I think it drives excitement. It gets people excited about the company, gets people excited about what we're doing. And really, that's what we need. We've been very Canadian, to be honest. Like probably like most of the world hasn't heard of us. And yet we're developing these really important drugs to reduce opioid use, right? That's a huge thing, and that's a huge societal benefit. We'll make the world a better place, and nobody's ever heard of us, right? So I think the LSO award is really good because it sort of helps to get the name out there, helps to promote AmacaThera, helps to promote what we're doing, and really validates what we're doing. So yeah, we're pretty thankful for LSO for that award.
So where do you go from here? I mean, what's the plan for 2026 and beyond?
Right now, I'd say a lot of this is heads down with Pacira. Super wonderful group. They've brought their team, we've got our team, we're working hand in hand to drive forwards, but it is a lot of work. Like we're manufacturing GMP product, and we're going to be running this clinical trial. So very, again, focused, right? Laser focused on getting that going. The other place that we're sort of focused on is the other pharma partnerships. So these other partners where we're taking their drugs, combining them with our gel. Really, if we can get some proof of concept data around those, we're hoping we'll get additional licenses on the platform. So it's a pretty cool opportunity because we can license it for pain, we can license it for various different indications. So really focused on delivering with Pacira's the number one priority, but then seeing if we can secure additional licenses.
It's great that more and more delivery options are being developed, each one useful in its own way. And AmacaThera's innovative approach is certainly going to make a difference, or its innovative approach if you're in North America. Next, it's kidneys. To say Hansa Biopharma is busy is a bit of an understatement, and 2026 is going to be a truly transformational year for the company and its drug Imlifidase, which is for treatment of highly sensitized adult kidney transplant patients with a positive crossmatch against an available deceased donor. Let's find out more from Hansa Biopharma's CEO, Renée Aguiar-Lucander. Hi Renée, great to have you on the podcast. And before we go into Imlifidase and why 2026 is so important, maybe you could tell us a bit about the company.
Sure. So Hansa Biopharma has a first-in-class platform of enzymes that cleave IgG incredibly efficiently and rapidly. And so this platform really kind of focuses on a couple of different indications. So we have a lead product that's approved in Europe, and we're in a review with the FDA with a PDUFA date of December this year in the US. So what this really is focused on are two kind of areas. One is desensitization and one is autoimmune disease. So desensitization is really for two kind of different patient groups. One is for transplant patients. So we've run a lot of clinical trials in kidney transplant, where there are about 15% of all patients on the wait list are classified as highly sensitized. That means they have a very compromised or challenging kind of antibody profile, which makes it very difficult for them to ever find natural matches of organs. So by reducing, by cleaving IgG, which is what this product does, very rapidly and very kind of significantly, you bring your IgG down to less than 5% in a couple of hours. So by doing that, you can actually allow for transplants with organs who are not a perfect match. And the other one is gene therapy, where again, a lot of gene therapy companies deliver AAV vectors. And so a lot of fair amount of patients often have antibodies against those. And so again, by rapidly reducing those AAV vector antibodies, we can basically allow that dosing to go through in terms of gene therapy. So that is really on the desensitization front, and there is really nothing else out there that does what this drug does today. And in the other area is autoimmune disease, which we're focusing on neurological autoimmune diseases, and we're starting with Guillain-Barré, which is called GBS, which is kind of a peripheral nervous system, IgG-mediated rare disease. So those are really the two areas that we're pursuing.
And I know that you've recently got to the company. Obviously, it's not like in the last month or so, but how are you finding it so far? And what was it that attracted you to Hansa in the first place?
Yeah, so I joined Hansa at the end of April last year. What attracted me to the company was really that, there is this kind of technology that the company's had for quite a long time and been developing it for these kind of applications. And it's really become quite clear that there's really no one else that's been managing to do anything similar in the industry. So this is still a very unique technology platform. And I think that the great value in this, really lies very much in the US market. A lot of it's driven by the national organ allocation system in the US and some structural kind of benefits over here, as well as really patient organizations, and that there is a really appreciated kind of high unmet medical need for these patients who are highly sensitized. So the combination of having a really unique platform, very significant US market opportunity, and, obviously complemented with an interesting pipeline as well with additional opportunities in rare disease, really was what attracted me to the business. The company was in need of restructuring. So I think there was quite a lot of work that needed to take place in order to position the company to actually be able to benefit from these opportunities. And I felt that my background was probably reasonably well adapted to being able to carry out that required restructuring and work and renegotiations in order to really kind of enable the company to benefit from these assets.
And speaking of those assets, you just had a press release came out within the last week about patients awaiting kidney transplantation. So I wonder if you could maybe tell me a little bit about Imlifidase and what it is, how it works.
Sure. So Imlifidase is basically, it's an enzyme that's been chemically designed to really focus on IgG. So what it does, it cleaves IgG in two places to really just completely make it inactive. And so it does that within a couple of hours. So between 2 to 6 hours from a 15-minute infusion, you will basically have reduced your IgG levels down to less than 5%. One could obviously then question, why would you want to remove your entire immune system that rapidly, and for what reason? And this is then when we come into this application in terms of highly sensitized patients. So there are, as I said, about 15% or so in the US on the wait list who are highly sensitized. It's basically defined as having what's called a CPRA score of above 80. That would mean that if you have a CPRA score of, let's say, 95, that gives you only a 5% chance of ever finding a matching organ. So this is kind of why a lot of these patients, they're on the wait list for transplant for many, many, many years. The median wait time for these highly sensitized patients above 80 is about 7 years. That would tell you that half of these patients wait longer than 7 years on the wait list. And obviously, these patients are on dialysis, and we also know that the 5-year survival rate on dialysis is somewhere between 40 and 50%. And so we know that every year, about 2,500 of these highly sensitized patients either die on the wait list or are moved off the wait list because they're too compromised to actually be transplanted. So there really is a very significant unmet medical need here. And what this drug can do is that you take one of these patients and you decide you're going to try and do a transplant for them, you can actually remove in the system, there's an automatic matching system, national kind of organ allocation. So you can take their profile and adjust it and remove some of the antigens to make them feel or seem less compromised in terms of an antibody profile. And that will then, over some period of time, hopefully get them an organ offer. They will improve their chances of ever getting an organ offer. And once you get that organ offer, most likely, because obviously you've done this in a synthetic way, that organ probably isn't a perfect match for this patient. And so what you can do is you can then, by giving imlifidase, you remove those antibodies, you can transplant this organ into this patient, and it'll be very low levels of antibodies for about 5 to 7 days. But obviously these antibodies will come back. Your body will start producing them again, obviously. But what happens is with all transplant patients is that post-transplant, you will give these patients autoimmune suppression. There is protocols for all transplant patients, irrespective of whether they're highly sensitized or not. So by doing that, you will keep those antibodies under control. And what we've shown is for a 5-year study, we followed patients for 5 years, is that the long-term performance is identical to whether you have a natural matched organ or whether you have gone through this type of unmatched, organ transplant utilizing imlifidase. And the reason, obviously, for this working is, in a transplant setting, you don't have a huge amount of time. And you really need to have something that is consistent, predictable, and safe for these patients in order to do this transplantation. Because if you do try to transplant an organ that is not sufficiently matched and you don't have desensitization in a patient, what will occur is what's called a hyperacute rejection. And so you cannot really, it's physically impossible nearly to actually do these type of transplants without controlling for the patient's antibody profile.
So what was the recent FDA announcement on this?
So the FDA basically provided us with a PDUFA date in December of 2026. So this is when we're now going to start a review period with the FDA. And so with a view to actually having that target PDUFA date in December of this year, which will then hopefully obviously lead to an approval of the drug. It would be an accelerated approval. That is the pathway under which this drug has been investigated. And so we would hope to be able to, subject to that approval, obviously launch the drug in the US first thing 2027. And so that's really exciting. We have already a senior team in the US and we're now building up those capabilities, supply chain, and again, mapping out of this whole market in front of us so that we will be ready for a commercial launch January 2027.
That's excellent news. Could you tell me a bit about the other things that you have, like your enzyme technology platform and what that addresses?
Sure. So the platform, obviously, apart from the kidney transplant, it also then addresses, for example, gene therapy. And this is a similar mode of action in terms of we have partnerships with gene therapy companies. So we have one with Sarepta, we have one with Genethon, and they're really exploring different AAV vectors, different tissues. And what we've been able to show last year is clinical data from both of those partnerships showing that imlifidase also does cleave, over 95% from baseline. So therefore, what that can do is it brings these patients into kind of either compliance with a label or with a clinical trial protocol, so that they otherwise would have been excluded from being able to be given this gene therapy. Because we now can do this, they can actually be included either in clinical trials or in a commercial setting. Because obviously, once the actual gene has been delivered to where it needs to be, the fact that antibodies will come back after a week or so is not really an issue, obviously, in the same way as it might be in transplant. So this is really kind of something where we will be looking to partner with other gene therapy companies. And thereby, really, a lot of these gene therapy indications are rare. And for a lot of those patients with AAV8, AAV9 vector kind of delivery systems in particular, there are a lot of patients, anywhere from 15 to 50%, that would potentially be excluded because of their antibody profile. So this, I think, is a very important help for all of those rare disease patients who otherwise would just not be able to benefit from a gene therapy. The second thing that obviously we have another enzyme off the same platform. It's called Hansa 5487. That is derived from a non-human host, in contrast to imlifidase. And so that enzyme is significantly less immunogenic. So what we have shown in a phase 1b study with this drug is that we can most likely redose with this particular enzyme due to the fact that you have a reduced number of ADAs that are produced by the drug because it's less immunogenic, and also the fact that it's very potent. So we could redose potentially with this drug after a 6-month period, because we've shown 6 months, 9 months, 12 months, 9 months and 12 months, it's really kind of 100%, success in terms of cleavage. And so I think this is a slightly different profile. And we've actually run a proof of concept study already in GBS with Imlifidase. So we know that there's very strong proof of concept in this rare disease. And so what we are doing now is we're actually going to have an interaction with the FDA and hope to basically come to an agreement with regards to a late stage clinical program for 5487. And so we hope to be able to have that information before mid-year this year. So that is another really kind of exciting franchise. And obviously in neurology, there are, apart from Guillain-Barré, there's also MOGAD and NMO and some other kind of IgG-mediated diseases. But we're certainly looking forward to kind of starting up a clinical trial, hopefully before the end of the year in GBS.
Do you have a bit of information on GBS?
Sure, absolutely. It's a rare, acute, paralyzing kind of inflammatory disease of the peripheral nervous system. And it is caused by this kind of immune system damaging nerve cells and structures. So you have this kind of rapid onset and progression of muscle weakness. It can lead to severe paralysis, arms or legs. And then you also obviously can be affecting your lungs. You'll have to go on ventilation. And so about 25% of patients require mechanical ventilation for days to months following this acute autoimmune attack. And it also results in the fact that about 20% of patients are still unable to walk after 6 months with today's kind of existing medication that's available. So this is really an IVIG standard of care, which is really being used today, but there are no FDA-approved treatments for this disease. So there is this kind of acute attack followed by a slow kind of recovery based on what's available today. So the treatment approach here is obviously for us to be able to, very rapidly kind of reduce these antibodies and thereby really significantly improving the functional status and the expedited muscle recovery. And we think in terms of the addressable population, we believe that there is about 15,000 patients in Europe and the US, and a quite significant incidence as well in that area. So there is quite a significant patient population and a significant unmet medical need here.
I know that you're getting close to that launch for Imlifidase and in different stages for other conditions, but could you tell me a bit about the clinical trials that you've either been involved with or are still ongoing?
Sure. So there was a phase 3 trial in the US that was ongoing for a long time, for several years, 2 to 3 years, because obviously finding these kidney patients, delisting their antigens, finding an organ, etc. was a pretty complex trial. But that trial read out in September of last year and was really, I think, pivotal for really kind of establishing the unmet medical need in this patient group. So this was 64 patients that were randomized 1-to-1. And so in the Imlifidase arm, and exactly what I was describing before is basically what happened. So patients were kind of recruited. These were patients that had a so-called CPRA score of 99.9 or above. So basically they had a probability of 0.1% to 0% of finding a natural match. So clearly highly compromised patients. And there are about 3,500 of those patients on the wait list today in the US. So what we actually then did is these patients were screened, and when they were then their antibody profiles were adjusted in order to allow them to get some organ offers. So all of these 64 patients had an organ offer at the time of randomization, and they were then randomized either to an active or a control arm. In the active arm, they could basically then check if the organ was a negative crossmatch or a positive crossmatch. The expectation would be that it would be a positive crossmatch because the organ would not be matched to the patient. And you would then basically give Imlifidase, do another test to ensure that that had turned to a negative crossmatch, and you would proceed with the transplantation. In the control arm, those patients and physicians had access to what else is basically available. So they could use IVIG or Plex or rituximab or cluzimab, a combination before, during, after, to their choice, according to any exploratory desensitization approaches that they would choose to potentially use. And what we saw during this trial is that the p-value of the trial ended up being 0.0001. That really shows you that, there's not really anything available today. There is no standard of care, and there's really nothing on the market today that can address this patient population in a way where both physician and patient would feel that it is consistent, predictable, and safe. So what we saw was obviously that out of the 32 patients in the control arm, only 3 patients actually decided to progress to transplantation at randomization, whilst in the Imlifidase arm, 27 patients progressed to transplantation at randomization. So very, very different. And the primary endpoint was actually then, what was the kidney function of these patients 12 months later? So not just being able to do the transplantation, but also looking at kidney function after 12 months from randomization. And what we saw then was that the mean of the Imlifidase group was 51.5 mils per minute, and the mean of the control group was 19.3 mils per minute. So that would also show you that obviously, even if you do manage to potentially transplant patients without kind of Imlifidase or with what's existing today, the outcomes may not be particularly good or helpful for those patients.
Is it something that you continue to, after the clinical trials, you continue monitoring?
Yes. So because this drug has gone under kind of what's called the accelerated pathway in the FDA, there's always a requirement for some kind of confirmatory data to be delivered. And what we are doing here is just really following these patients for longer term. So we will have an obligation to try and follow these patients for basically 4 years post the primary endpoint. So a total of 5 years. And so this is basically to ensure that this has long-term clinical benefit. The company has already, with all of the phase 2 patients that went through the transplant, have followed those patients on a pooled basis and looked at 5-year outcomes. And this is why, we have actually shown that it's virtually identical to a normal kind of non-highly sensitized transplant outcome. But that is basically, and it's highly likely because they are in a variety of nephrology indications. And you can look at this, and it's been quite a lot of papers published on this in a variety of areas, is that 12-month eGFR data has shown to be very predictable of long-term outcomes. So we feel very comfortable with the fact that, having such strong 12-month eGFR data, we think that data is and will be highly predictable of long-term outcomes in these patients.
Are there other applications for Imlifidase?
Yes, well, I think that, we are obviously getting quite a lot of requests in terms of compassionate use, et cetera, from other transplant settings of solid organs. So obviously, heart, lung, liver transplants, stem cell transplants. These are all areas where I think there's an interest and an unmet medical need that we could potentially look at. Obviously, the first thing we have to do is to get approval in kidney transplants, and then we can actually potentially move into other solid organs or other transplant settings. And obviously, there could also be an application for this drug in antibody-mediated rejection, which really is, that happens to patients who are, not highly sensitized as well, obviously. And in those settings, the company has done some earlier investigator-led trials that had, shown that could potentially also be an interesting application. So I think that there are definitely things. I think we're trying to take one thing at a time. And obviously the market opportunity is very, very big in kidney. Our greatest focus right now is obviously getting it approved and getting it out to patients who are on the waiting list, and many of whom will die, unfortunately, if we don't get to market. There is another trial as well that's ongoing, which is in Europe. So there is a phase 3 trial ongoing in Europe, which has yet to read out. That will read out in the middle of this year. That is 50 patients which have been transplanted, highly sensitized, across 23 sites in Europe. And that will add, obviously, to the clinical information and to the data that we have on this drug. That was fully recruited in March of last year. So we are expecting it to read out mid this year. So those are the kind of clinical trials that are kind of ongoing. In addition, obviously, we have a pediatric trial, which is a regulatory kind of requirement from EMA. And as I've mentioned before, the GBS, which is something that we would hope to start before the year end.
And you mentioned about getting to market with Imlifidase. is that going to be done independently, or do you have partnerships in place or looking for partnerships?
So in Europe, the company has chosen to go to market themselves. So we do have our own commercial sales force, et cetera, set up in Europe. And in the US, we are also really definitely considering or planning to go on our own in the US. This is a very tight call point. It's a very focused sales effort because there's only about 200 hospitals in the US who actually conduct kidney transplants. And out of those 200, 100 of them really represent about 80% of the overall volume in the US. So in order for us to commercialize, we really only need 15 to 20 salespeople. Our total US business might be 40 to 50 people. So I look at this as a very kind of manageable investment for us, and also something where I would expect that this would be able to be cash flow positive within a fairly short period of time relative to many other launches of drugs in the US.
And I would assume that because of what you're doing and the relatively unmet medical need surrounding that, that awareness and the selling point, you don't have to really promote it that much.
It's not going to be heavy promotional. It's quite medically driven. It's really kind of more about having medical affairs interactions, explaining to the team at the hospital, which includes both the HLA director, the nephrologist, the transplant surgeon, pharmacist. I mean, there's always a whole team of people that you need to make sure that you communicate with. But it's really to explain the mode of action of the drug and share the clinical data that's available, and really try to do that as much as we can with these teams in the hospital. So it's not really a heavy promotional product that way, but it is much more about scientific exchange and that kind of medical affairs explanation. And quite a lot of this will also be driven, obviously, then by information and data that we can share at congresses or in manuscripts and posters, etc. Because this is really a group of patients that has been classified as obviously being very, very difficult and, with very limited success in terms of trying to do something for these patients. This will be kind of a change in how physicians look at this patient group. And so I think that's why it's so exciting for us that we can really bring something to market that we truly believe is going to, really transform the lives of these patients.
And you mentioned you're over in the US now and there are lots of conferences. What's reaction like at those conferences?
I think that it's very positive. I think that, the data is very compelling. I mean, having these kind of clinical trials, which I think people appreciate, this is a complex trial. This was not a simple design. And I think everybody, even realized the fact that, that control arm was very encompassing in terms of allowing lots of different, things to be applied. So the fact that we had such an incredibly strong p-value coming out of that trial, I think really underscores the fact that there really isn't anything else there that really can provide this solution today. So I think that there is a lot of interest and support. I think that, as always in these situations, and maybe particularly now, I think that, there is an FDA process to go through. We're just going to have to get through that review process. I feel very confident about this process. I think the clinical data is very strong, but I think that everyone acknowledges that this is a very significant market opportunity as well. And so I think there's excitement around, the investor meetings. And we also have quite a lot of kind of catalysts coming out this year, because obviously we have another phase 3 trial reading out in Europe and another filing to generate in Europe. And there's also a lot of interest around kind of autoimmune diseases and GBS. So I think that we have a lot of things to talk to them about. And so far, so good.
When you're doing clinical trials, phase 1, phase 2, you're building that momentum, but now you're getting into an entirely new phase of the company. How are you managing those expectations and that transition?
It is a big transition, actually, and particularly for a smaller biotech company. It is a bit of a cultural shift, going from kind of a very R&D focused kind of setting to more of an integrated perception where actually, yes, we will do discovery and R&D, et cetera, but we also are going to have a significant commercial presence in the US now as well. So I think that there is absolutely a transition, and I've worked very hard at kind of since I came in to really try to convey what this is going to look like and what is going to be required and how do we do this in the best way. And this is really balancing resources and capital and skill sets internally, whilst also obviously complementing them, which is a necessity, with new skill coming from the outside. And so I think what I've done since I came in is obviously we've had a revision really of kind of like, what is this company's vision, mission, what are the value words that we really need to take seriously and really live by every day. So really kind of engaging the entire organization in this process. And then also obviously bringing in people with significant expertise, both in the clinical setting, but as well in the commercial setting in the US, and making sure that the organization can see what those skills are, why they're needed, what we're doing. And so I think creating transparency, also focusing a lot on accountability, making sure that we retain our agility as a small biotech company, but actually just build on some of these core pillars in order to ensure that, we will have not just a very successful launch in the US, but that we also therefore continue to build our pipeline, retain our discovery, do all of those other things in that value chain simultaneously. It is very rewarding, and I think it'll be incredibly rewarding, once we actually hopefully can bring this to patients in the US as well. And also really patients who today have extremely limited options, which is always, I think, extremely rewarding to be able to contribute to that.
So, more great work, and with results from one phase 3 clinical trial later in the year, and hopefully positive news on Imlifidase approval, the company will be in a whole different position a year from now. So that's it for this week's podcast. 3 great guests and me. If you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact us at jim@deeptechdigest.com. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.